In this last segment, we will talk in a little bit more detail about other fMRI techniques. So you mentioned that once you have the activation pattern in a particular area, how do you actually know what to do with that or how to analyze it?
Right, so I mentioned there's the most standard technique is a subtraction technique with fMRI, where you get, you're comparing the response of the brain in two separate states, say a 10 left or a 10 right. And the way you analyze the data from that kind of experiment is you take the images that are associated with a 10 left, or one condition, average those together, and you take the data associated with the other condition, those temporal frames. So now I've got two sets of images. I simply take the difference between those guys. And those differences, any big differences will be associated with a difference in the response between those conditions. And that subtracts out any kind of low frequency drift and things like that that we often see in our voxels.
So essentially you have a baseline condition and then you have the actual experimental condition, and you're just taking the baseline activity and subtracting, or vice versa, the
That's
activity.
Right. We always have to compare two different states. That's the thing. And the reason for that is that if you look at the raw data from a given voxel, if you look at the image intensity over, say, a 6-minute scan of a subject just lying in the dark doing nothing, this voxel will do all sorts of crazy stuff. There's all these low frequency drifts, and sometimes it'll drift up, sometimes it'll drift down. Its neighboring voxel will do something completely different. We don't really know why, and it really looks like it's the major component of our data, but what we in fact do is just simply subtract that out, subtract out the main trend, and again do the subtraction technique, because these low frequency drifts are things we're not interested in, so we just get rid of it.
Other than the subtraction technique, what other methods are used to make the data set meaningful?
So ideally, we'd like to be able to present more than just two different stimuli, or put people in two different states when we run a scan. I'd like to present people all sorts of different stuff and measure the response to these individual things. And more recently, there's been designs called event-related designs which allow us to do this. So the idea is you present, say, a house and a car thing every couple seconds, present different stimuli. And it turns out that we can actually measure the individual responses to these individual events. It relies on this interesting fact that even though the fMRI response is really, really slow, it peaks 5 seconds after the stimulus, I can present a second stimulus just a couple seconds after the first one. It'll produce its own response that simply adds on top of the previous one.
Wouldn't there be interference?
Well, you'd think so. In fact, there might be some, but there's surprisingly little. What we're dealing with is what we might think is a somewhat linear system, where the responses add together. It's an ideal setup, because if the system is linear, then we can present any complicated image sequence, and even though the responses are overlapping over time, we can do what's called a deconvolution technique and estimate the response to any individual event. But it's surprising, because as we mentioned before, we know so little about this coupling between the neural response and these blood flow changes. Why should they just simply add on top of each other? We don't know. You'd think they'd maybe saturate and compress. Who knows? But it just seems to work remarkably well.
That's interesting. Is that true for the entire brain, or is it just for the visual cortex?
Well, we don't really know, because what we really want to know is the relationship between the neural response and the fMRI response. But of course, we don't know the neural response. That's why we have jobs. So what we really look at is the relationship between the stimulus and the response. And those things seem to add nicely, but we don't really know the direct relationship. Although there's more and more effort trying to put monkeys inside the scanner, trying to do a simultaneous electrophysiology in monkeys and fMRI to try to really test this linear systems hypothesis. And it's reasonably good.
You talk about fMRI adaptation in one of your articles, actually several of your articles. What is that precisely?
Well, so fMRI has good resolution, spatial resolution, like I mentioned, on the order of a millimeter. But as for a physiologist, that's horrible, because neurons are, very, very, very small. In a given fMRI voxel that might be, say, 2 by 2 by 4 millimeters in size, there will be literally millions of neurons inside of there. And so we're very far away from measuring the response of individual neurons. So, for example, if I present stimuli of different orientations and measure the response in V1, a given V1 voxel probably won't change its response very much, because it has neurons with all sorts of different orientations in there. Adaptation is a classic trick of trying to get at these subpopulations of neurons. It's been used in behavioral psychophysical experiments for over a century. If I present a stimulus that makes a neuron get excited, I present a stimulus later, that neuron will respond less than it did before. It's somehow adapting or fatiguing or wearing out, whatever term you want to use, responds less subsequently. So, if I suppose I've got a V1 neuron that has all sorts of different, a voxel inside area V1 with fMRI, and it represents all sorts of different orientations. If I have a subject stare at a vertical thing for a long time, and then go ahead and measure that voxel's response to different orientations, I will see a difference. And that's because the subset of voxels that prefer vertical orientations are going to be adapted. And so they'll respond, the voxel will respond more to horizontal than vertical. Mm-hmm. And so that can be a trick for getting evidence of tuning or selectivity of neurons within that given voxel, and getting around this problem that this voxel represents all different directions or orientations or colors or whatever stimulus dimension you're messing around with.
That's interesting. Are there any confounding variables or other things such as inhibition that kind of make it hard to look at these subpopulations?
Well, so after these adaptation techniques originally developed to study responses in the lateral-occipital cortex, this region of the brain that responds to objects and things like that. And it's amazing is that if you present a face to a subject, an individual face to a subject, and you get a response in this region of the brain called the fusiform face area, you get this big response. If I present with fMRI response, if I present the same face again, the response will be only like half as big as originally was, even though the stimulus was only presented for like a half a second.
You actually adapt to faces?
Yeah, exactly. There's huge adaptation effects to faces. I found this really surprising because primary visual cortex neurons don't adapt like that. In order to make a neuron fatigue in early visual areas, you need to really hammer on it. In the psychophysics labs, we sit there people have to look at horizontal things for 30 seconds before it gets harder to see a horizontal thing. And it turns out that V1 doesn't adapt like the lateral-occipital cortex. So if I present a vertical thing for half a second and I present a vertical thing again, the change isn't anything as drastic as you see in the lateral-occipital cortex. So the strength of adaptation varies throughout the brain, and that's a problem for the technique. Because if we're using adaptation as a tool, and that tool varies in strength across the brain, then the strength of our results are going to vary in very complicated ways.
What happens when you have multiple properties that you're looking at once, or multiple visual features? Does that complicate your analysis?
Well, so ideally we'd like to be able, like I said, I can measure the response to neurons that prefer this particular location in the visual field. And the reason for that is we have these retinotopic maps that are nicely laid out, and so spatial location is really nicely coarsely encoded in a way that is accessible with traditional fMRI. But features like color or orientation or direction are represented in clumps of neurons, these columns, right? Hubel and Wiesel orientation columns that are too small really to pick up directly with fMRI. And so we have to use tricks like adaptation to try to get at that. And there's a newer technique, these so-called pattern classification, multi-voxel pattern classification algorithms that are trying to do the same sort of thing, to try to get at representations of features like orientation that are represented at a resolution that is too small to be measured with normal fMRI.
Mm-hmm. So you can actually tease apart, like, you can figure out how many neurons in a specific area code for motion or color or other visual features using these techniques? Kind of.
I mean, the idea is that even though a given voxel will have, say, all orientations represented in there in V1, because the coarseness of these orientation columns are on the order of maybe half a millimeter, and our voxels are 2 or 3 millimeters, you might actually have a slight bias in the representation of this voxel from one orientation or another. So if I were actually physically change the orientation of a stimulus across different trials in an fMRI experiment, I actually will see a subtle difference in the strength and the response in this voxel. In the past, we thought there was nothing to that. There's just too weak to be able to do anything with it. But if you look at this pattern of responses across all voxels, maybe hundreds of voxels in area V1, it turns out that each voxel will change its response slightly differently to each different orientation. And if I use all of these voxels together and actually think of the response to each orientation as a separate pattern of responses, there's actually a lot of information in there. So for example, I can have subject lie in the scanner, and I can measure the response across V1 to different orientations. And I get, say, 8 data sets for 8 different orientations. This is V1 responding to these 8 orientations. Then I present a new orientation to the subject, and I get a new pattern. And I can compare this pattern to each of these 8 patterns that I measured before. And I say, which one does this match most closely with? Turns out that I can do very, very well. The pattern that will match most closely with will be the orientation that I used to measure that pattern before. So I can actually predict what orientation was presented to that subject, which is surprising because we didn't think we knew anything about orientation.
Can you actually predict what they're thinking about too?
Right. So this is a classic study now by Kamatani and Tong, which did that experiment. First, they did the classification for physical stimulus, and they were able to say, okay, I can predict what stimulus they saw based on this training set. Now what they did is they presented a plaid stimulus that had two orientations at the same time. And they had subjects that tend to one orientation and ignore the other one. Okay. Turns out when they did that and compared the response to that condition to these patterns, they were able to, this pattern matched more closely to the vertical stimulus than the horizontal one. So in other words, attending to vertical orientation, ignoring the horizontal ones, made the pattern of responses in V1 look like there was just a vertical stimulus on the screen. And they could predict that with fMRI. So in a sense, they were mind reading. They were able to guess which orientation the subject was intending to and which ones they were ignoring.
Are there other instances in which you can use fMRI to infer what somebody's thinking about?
Yeah, so that was a kind of a landmark study. And people have used these pattern techniques in all sorts of areas now, outside the visual cortex, or in the visual cortex in the area MT, for example. So we've been able to show that you can predict which direction of motion a subject is responding to or paying attention to in area MT and V1, turns out. But you can do it across the whole brain. If you take all the voxels in the brain and treat that as one big pattern, people are starting to play some fun games, like you can actually predict what category of thing people are thinking about. Are they thinking about, are they trying to recall things, or are they trying to store memories, or are they using mental imagery? You can actually accurately predict what a subject is doing, what kind of brain state they're in using these techniques.
That's very interesting.
Yeah, it's a big deal right now.
Well, I think that about wraps it up. Thank you for taking the time to interview with us, and that was a very informative and interesting. The whole thing was.
Well, thanks. It's been fun.